Hello, everyone. Thanks to Arm for the opportunity to present the TYNA story today, and thanks to all of you for coming here to listen to the story. Make sure I got this right. We are a public company. I'll be making forward-looking statements. These are some of our standard disclaimers. First and foremost, what makes Tenaya unique is our mission. The previous two speakers talked about platform technologies with broad applicability in multiple therapeutic areas, and we at Tenaya decided to do something different. We decided to focus on the heart, the leading cause of death in the world, more than all oncology combined. And something else that makes us unique is we decided to do drug discovery in a modality-agnostic manner, which means we actually internalize multiple modalities, including gene therapies, genetic therapies, which I'll focus my talk on today, but also cellular regeneration approaches and even small molecules. So we go after both rare and prevalent forms of heart disease. We've chosen, our mission is to discover, deliver, curative therapies that can address the underlying causes of particularly genetic forms of heart disease. We have internalized research, and I'll talk a little bit about some of the unique things we've done over there. We're now a clinical stage company and building a development organization. We've also fully internalized manufacturing, which I'll also talk about today. We've achieved all of our milestones that we've set out for this year with one left, and we're on track to file an IND for TN-201 before the end of the year. That's the leading genetic cause of hypertrophic cardiomyopathy, which I'll talk a little bit more about today. So a couple additional things that make Tenaya unique is I mentioned, we're focused on the heart, modality-agnostic drug discovery, but we don't have one platform. We actually have three distinct platforms that we do to solve different problems that have historically plagued the field of drug development for heart disease. Gene therapy platform, cellular regeneration, and precision medicine. In the interest of time, I can't really go into all of it today, but we'll focus on gene therapy. So gene therapy, the problem is to improve heart cells that are defective usually because of a genetic defect. And we can use this to do gene transfer today, vectorize a therapeutic protein in the future, also do gene editing with this platform. Cellular regeneration is trying to solve a fundamentally different problem. These are not heart cells that are defective, but heart cells that are lost, usually because of an ischemic event. And then our precision medicine platform, we're actually using human iPSC-derived cardiomyocytes to do phenotypic screening in human cells, identify targets of interest, and then do drug discovery in a modality-agnostic way. We've chosen to internalize the capabilities, particularly related to genetic therapies. So we've internalized capsid engineering. A lot of the work that David at 4DMT have done inspired us, and we've built on that with a focus on the heart. Also doing engineering of promoter and regulatory elements. We also explore different drug delivery mechanisms, direct to the heart, infusion IV versus infusions closer to the heart, and also AAV manufacturing. We've chosen to internalize all of those capabilities in addition to in vitro and in vivo disease models. So quite a lot going on under one roof for our company. Here's our pipeline. Our more advanced pipeline is on the top in the orange, and then a very, very long tail of things going on beneath the surface. Again, all focused on both rare and prevalent forms of heart disease. Actually, the lead program, just to the spirit of being a modality-agnostic company, our lead program, TN-301, is a small molecule for a large indication, heart failure with preserved ejection fraction. That IND is cleared. We're dosing patients in a healthy human volunteer study. Because of the nature of the conference we're at, I won't really speak about that one today. But we're excited to be a clinical stage company and be able to share data on this program next year. What I will talk about today are TN-201 and TN-401. TN-201, both AAV gene therapies, both lock and key gene therapies addressing the underlying genetic defect. TN-201 is doing that for hypertrophic cardiomyopathy. TN-401 is doing that for arrhythmogenic cardiomyopathy. I'd like to point you to the epidemiology column. We're talking about large orphan conditions. In one case, more than 115,000 patients in the US alone, and in the other case, more than 70,000 patients in the US alone. We wholly own everything in our pipeline. There we go. So first, TN-201. With TN-201, what we're addressing is the leading genetic cause of hypertrophic cardiomyopathy, and that is due to the MYBPC3 mutation. It's a mouthful. It affects about 20% of all HCM, so that's how we get to the epidemiology number. This is a severe disease. These patients present with enlarged hearts, thickened ventricles, arrhythmia, sudden cardiac death, definitely reduction in their heart function. Eventually, many of them progress to heart transplant. There's early mortality associated with the disease, particularly in infants. The patients who have two genetic mutations don't survive past the first few weeks or months of life. So it's a pretty severe need and a lot of unmet need. There are no therapies that address the underlying genetic cause of this disease. This is the disease of the sarcomere, you can see the little pictorial with the MyBC3 protein connecting the thin and thick filament in the sarcomere. We know that the patients on average have 60 to 70% of the wild-type protein as a result of their mutation, and so our goal with TN-201 is to add a full-length copy of the human gene into the cardiomyocytes using an AAV9 vector and express the MyBC3 protein and get them closer to 100% of wild type. Our reason to believe that this can work comes from the animal model. This is the preclinical model, is an ultra severe double homozygous knockout model. And I'll call your attention to the graph in the middle. Blue are the untreated animals, and orange are the treated animals. The blue animals rapidly progress. Pretty much at birth they have disease, and they eventually die within the first year of life. And after a single administration of TN-201, we see a reversal of the disease, an improvement in ejection fraction that is quite profound, 20 to 30% improvement in ejection fraction, which if we saw that in humans would be remarkable. And then you can see stability of that effect going out. We took these animals out to their natural lifespan with about 20 months. By the end of that study, 100% of the untreated animals had died, which you can see in blue. 100% of the treated animals had survived. And I mentioned that in humans, they're missing 30 to 40% of the wild-type protein in these animals, and in this experiment, we produced 100% of the wild-type protein. So that hopefully gives us hope that if we can achieve that in the clinic, we can do something good for patients. The other disease I'm going to speak about is, the program is TN-401. Over here, we're dealing with the leading genetic cause of arrhythmogenic cardiomyopathy, and this is due to the PKP2 gene. This accounts for about 40% of all ARVC, so that's how we get to our number, about more than 70,000 patients in the US alone. This is a disease not of the sarcomere, but a disease of the desmosome. Desmosomes are the structures that hold cardiomyocytes together. So they both electrically and mechanically allow the cardiomyocytes to couple together. As a result of the missing protein, these cells kind of start coming apart. That affects heart rate, that affects arrhythmia, which is one of the dominant symptomologies in this disease. And in fact, unfortunately, for many children and young adults, the initial presentation of this disease is sudden cardiac arrest and sudden cardiac death. You can see the average age of mean cardiac arrest is 25 years old. So it is a severe disease, and again, a lot of unmet need. There are no therapies that address the underlying genetic cause that is causing this form of heart disease. TN-401, another lock and key gene therapy, we're delivering a full-length copy of the PKP2 gene to the cardiomyocytes using an AAV vector, and with that express the wild-type protein and hopefully have a beneficial effect. Again, we have very strong preclinical data supporting this program. Sometimes the visuals help more than numbers. At the top, you see a normal sinus rhythm of a wild-type animal. In the central panel, you see the abnormal electrocardiogram from an animal that has the PKP2 mutation. There are a number of things that are going wrong in this animal, and then a single administration of TN-401, you can see a return to the normal sinus rhythm. We were actually also to quantify that with something we call the ventricular arrhythmia score, and you can see wild type, a very high number of the control animals, and with gene therapy, a near reversal of the arrhythmia. We also see benefit in other aspects of the heart. We see a prevention of the enlargement of the right ventricle, which is where the name of this, arrhythmogenic right ventricular cardiomyopathy. So we see prevention of that cardiomyopathy. We see an improvement of the left ventricular ejection fraction in a dose-dependent manner, and we see a survival benefit in this program as well. So two exciting lock and key gene therapy programs, both using AAV to deliver a healthy copy of the gene with very, very compelling preclinical data supporting our transition to the clinic. I did mention earlier that we have chosen to internalize core capabilities, so in my closing minutes, I'll talk about two of them, AAV capsid engineering as well as manufacturing. So on the capsid engineering, in some ways, the 4DMT talk that preceded us laid it out for the audience exactly how that works. We're doing something very similar, but with an intense focus on the heart. So we have synthesized and at this point screened more than 1 billion variants, all intended to identify novel capsids for the heart that outperform the gold standard, which most people would agree is AAV9. We've done this screening in human cells, in mice, in NHPs, multiple rounds of screening in NHPs. We even just to be thorough, test pigs, because a lot of drug discovery in the heart has to go through the path of large animal studies in the pigs. So we're looking very thoroughly across multiple species. We're looking for multiple attributes: improvement in transduction of the heart, improvement in detargeting of the liver, antigenicity, as well as manufacturability. So we're putting a very high bar for ourselves. These are data that are in the public domain, and we're showing not one, but multiple capsids that outperform AAV9 in terms of both transduction of the heart as well as detargeting of the liver. This is after 2 rounds of screening in NHPs. We've actually now have data from a 3rd round of screening in NHPs, as well as data in multiple species. We're going to actually, this week at ESGCT, we'll have a poster. We'll be giving an update on this efforts. We've been at this for 3 years, and we're going to continue working at this. This is clearly an area of strength for us. We believe that this sets us up to have not one, but multiple capsids to address different forms of heart disease that we can take into future programs. And with that, maybe reduce the dose at which we can have a therapeutic effect. Manufacturing is something that we know from the field. It's the Achilles heel of this field, and we had to take it seriously, and so we did. We have internalized both non-GMP and GMP capabilities, everything from shake flask up to vector core at the 50-liter scale, pilot plant operation at the 200-liter scale, and then this year we announced that we had completed the construction of our own cGMP facility and are producing drug as we speak at the 1,000-liter scale. In addition, we have actually internalized both HEK and SF9, and we have done improvements in both platforms to get to products that have higher, a production process with higher yields and better empty to full capsid ratios. We've actually filed IP on some of these developments. So we intend to make manufacturing a core area of strength. Why did we do that? Partly because we're doing work on novel capsids, and we felt that we needed to really control our destiny in terms of the process upstream, downstream, quality control, analytical development on our novel capsids, but also because heart disease is a large category, and we expect to go beyond just rare diseases. In fact, those 2 indications I mentioned earlier are pretty large orphans, and we intend to go larger than that. That means we need to be able to scale to ever higher, for larger indications, to even larger bioreactors. And so for that purposes, we have invested in GMP manufacturing capabilities so that we can actually bring down the cost of goods, produce this material reliably, and control our own destiny in terms of quality, cost, and timelines. So in close, Tenaya's got a lot going on, focus on heart disease, rare and prevalent forms, both gene therapies, in the future gene editing, cellular regeneration. We didn't have time to cover it all today. We've hit all our milestones in the first half of the year. The most important milestone that remains for us in the second half of the year is filing the IND for TN201, and we are very much on track with that. We'll file the IND for TN401 next year and initiate clinical studies for that. Our small molecule is already in the clinic, and we expect to present data on that in 2023. Importantly, in the current environment, cash is important, and so according to our public statements, we had about $180 million in cash as of the end of the second quarter of this year. That gets us at least into the second half of 2023, and we'll be giving updated cash guidance and clinical catalyst guidance at the beginning of next year. So we're well positioned to advance not only the 2 programs we mentioned, but all of the work, clinical and preclinical, that are happening, in addition to doing the platform enhancements, which we think will position us to be the leading precision medicine cardiology company. Thanks.